Good afternoon. Today we are going to get lost in translation. How to use that. This was you were? Yeah. This is the first, the movie was second after this. We're going to talk about how you make proteins from messenger RNAs. Remember that messenger RNA, which is central to all those genomes. We're going to talk about how that's made into protein or translated into protein. In particular, how viruses manipulate the system to do what they want. So we're going to review a little bit of translation first, just to make sure we are all at the same level, and then we'll move on to what the viruses do. So in eukaryotic cells, this is a typical messenger RNA at the upper right. It is a messenger RNA encoding an open reading frame, which is translated to make one protein. And this typical eukaryotic mRNA has a 5' cap structure, usually an untranslated region at the 5 and 3' ends, and a poly A sequence at the 3' end. And the cap structure looks like this. It's a very interesting linkage. Instead of a 5 to 3' linkage, which is typical of the bases in the mRNA or in DNA, a 5 to 3' linkage. There's the phosphate bond between the 3' end of this base and the 5' end of this base. The cap is actually a guanosine and a sugar linked 5' to 5'. So it's inverted. Normally this would be linked 3', but it's a cap structure. It's different. There are 3 phosphates in there. They're linked 5 to 5'. And this cap is found on most eukaryotic mRNAs with a few exceptions. And in fact, some viral RNAs, mRNAs, do not have caps. We'll talk about some of those. And it's often methylated, and that's important for its function. It has a variety of roles. It has an important role in translation, as we will see today. But it also has roles in processing of mRNAs, transport, mRNA turnover. And today we'll talk about the need for it for efficient translation. There are untranslated regions in the mRNA, and these will factor in our discussion today. There's a 5' untranslated region. It's usually about 50 to 70 nucleotides, the average on most cellular mRNAs, but it can be very short or even over 1,000 bases. And we'll talk about the implications of that. It often has secondary structures, those hairpins and stem loops and so forth in the RNA. And those structures would normally impede the ribosome. As we will see, the ribosome has to travel from the cap to the first AUG. And if you have secondary structure, that will impede the passage of the ribosome. So the amount of secondary structure in this 5' UTR will affect the efficiency of translation of the messenger RNA. It's followed by the open reading frame, of course, begins with an AUG and then ends with a stop codon. And then we have a 3' untranslated region, which also has roles in regulating translation, efficiency, and also stability of the mRNA. And the poly A tail here, which is necessary for efficient translation. The mRNA, of course, is translated by a machinery that includes ribosomes. And here's the composition of a eukaryotic ribosome. This is the entire ribosome. It sediments at 80S in a sucrose gradient. And it is made of 2 subunits, the 60S and the 40S subunits. And those, of course, are made of both RNA and proteins. A couple of RNAs for the big subunit and one for the small, and 30 and 50 different proteins. And this is the structure of the ribosome. It was solved a number of years ago. In fact, Nobel Prizes have been awarded for solving the crystal structure of the ribosome. It's an amazing feat because these are big assemblies, big macromolecular assemblies. We now know how they look, and that's what this diagram on the right is derived from. In this picture, the RNA is pink and yellow, and the protein is blue. In fact, you could take away all the protein, and what's left, the RNA, can still make proteins. It can still catalyze the peptide bond. So the RNA is actually probably is most important for making proteins. And this may be a relic of the RNA world, when there was just RNA and no protein, and these may have evolved way back then. There's also tRNAs, and we will talk about tRNAs a bit, so let's make sure we understand them. These are shorter RNAs whose job it is to transfer an amino acid into the ribosome during protein synthesis. And there's one, of course, for each amino acid, sometimes more than one. And there is a part that recognizes the mRNA, the anticodon, and then there's a part where the amino acid is attached. And these have secondary structures as well, as you can see here. These are highly structured, and you can see that here the anticodon is down here. It's an L-shaped structure with an amino acid attachment site up here. And these are examples of phenylalanine tRNAs. So this would be the codon for phenylalanine. Phe would be linked up here, and aspartate would be here as well. So tRNA. So the translational machinery, in addition to ribosomes and tRNAs, in eukaryotes consists of initiation proteins. We call those EIFs. Elongation proteins, which are called EEFs. And termination proteins, which are called ERFs. For some reason, they're not called ETFs. Do we know why? Sorry? What?
It's a business term.
Oh, okay, ETF. Anyway, these are ERFs, so you have to remember that. So let's talk about initiation, which is the step at which most of the regulation occurs in translation, which is not to say that it doesn't occur at other steps, but it's the best understood. There are two main ways that ribosomes bind to eukaryotic mRNA. There's the one called 5' dependent initiation, which happens probably on most mRNAs in cells. The initiation complex binds to the cap and moves down to the initiation codon, which can be an AUG, but it can be other codons as well. That was the one we knew of for many years until viruses revealed the second one, which is internal ribosome entry. And this is where the initiation complex binds just upstream of the initiation codon. It doesn't need a cap to work. The ribosome can bind internally, hence the name internal ribosome entry. So that was discovered with viruses. In fact, it was discovered with poliovirus and really changed our understanding of how translation works. So let's take a look at the first mechanism, the 5' end-dependent initiation. Some people call this cap-dependent, which isn't perfect. I think 5' end-dependent is better. And this depends on having an mRNA with a 5' cap, which is down here. It's the little blue box. So what we need to do is bring the ribosome to the mRNA and eventually to the AUG. So we start with a 40S subunit, and this 40S subunit binds a number of initiation factors. EIF3 in particular is very important. And then the ternary complex comes in. The ternary complex consists of the tRNA with the methionine on it. That's the initiator tRNA. EIF2 with GTP. That's the complex. tRNA, EIF2, GTP. And then another factor, EIF1, comes in as well. And that's placed onto the growing complex here. This becomes a 43S pre-initiation complex. Another protein comes in called EIF4G. That's the red one, and binds this as well. And the red one will in turn bind EIF4E. Now, EIF4E is this curved purplish molecule here, which is actually the cap-binding protein. This protein, EIF4E, has the ability to bind the cap. We don't actually know the sequence of events, the precise sequence of events here. It doesn't really matter. What you see happens is that EIF4E binds the cap. In turn, EIF4E binds to EIF4G. We'll look at that more carefully in a moment. EIF4G is binding EIF3, which in turn binds the 40S subunit. So you see we have a complex of protein-protein interactions which brings the 40S subunit to the 5' end of the mRNA. That's why we need a cap structure in order to enable this. In the process, we also have the ternary complex in here. So here there's a initiator tRNA ready to start translation, because remember the MET is most of the time the first amino acid which is put down. And then this has to scan and find the AUG. So we do believe that this complex in some way moves to the AUG. We don't know how that happens. We don't know when the cap is let go or if the RNA is threaded through this. This is an unsolved problem. But that complex does have to get down to the AUG. Now, EIF4G is a very interesting protein. It's large, 220,000 Daltons, And it's shown in a linear diagram up here. It has binding sites for many different proteins. It has a binding site for EIF4A, EIF3, EIF4E, and poly-A binding protein. And this is the way EIF4G orchestrates the interaction of the 40S subunit with the cap. So EIF4E is the cap binding protein. It has a binding site at the N terminus of EIF4G. It's right there. And 4G also binds EIF3, and EIF3 in turn binds the 40S. EIF4A is an RNA helicase. And this is very important to have here in this complex, because if you have secondary structure in the 5' UTR, it has to be unwound by this RNA helicase. So an RNA helicase will unwind double-stranded RNA. So if you have a lot of such structure, you need to have this EIF4A present. So here's a summary of the next few steps. We've made our initiation complex on the mRNA. And remember, if there is secondary structure, which is diagrammed here, you've got to have that EIF4A to unwind it. This is an ATP-dependent helicase. So in other words, unwinding this RNA requires energy. So you get energy hydrolysis as the complex moves down the mRNA. And eventually, it will reach that AUG codon. And then the met tRNA will base pair and start translation. When this AUG is reached, a lot of these factors are released. You can see here we don't need them anymore. The EIF3, for example, is released. EIF4G is gone. And also the factors associated with the ternary complex come off. EIF2 comes off. And in the process of this met tRNA sitting down, getting ready to translate, this GTP is hydrolyzed to GDP. So this placement of the met tRNA in the AUG codon requires energy. And that's important, we'll see later on. So try and remember that this ternary complex comes apart. It leaves, of course, the tRNA behind, but hydrolysis of GTP is needed. Once this happens, the large subunit can then come in and associate with the 40S subunit. And now we're ready to begin protein synthesis. We have a met tRNA sitting in the P site of the ribosome, and now we can put a new amino acid in the A site. Now, we also think that the 3' end is involved in this translation. There is a protein in cells called poly-A binding protein, PAB1P. It binds poly-A sequences, and it also binds the N terminus of EIF4G. A few slides back, there was a diagram of binding sites for other proteins on EIF4G, and one of them is PAB, poly-A binding protein. And there's quite a bit of evidence which suggests that the RNA ends have to be brought together by these interactions in order to get maximally efficient translation. So there's genetic and biochemical evidence. Unfortunately, no one has actually visualized in a cell a circularized mRNA. But there is a lot of other evidence which suggests that this is important for efficient translation. Why it is so is not clear. One idea is that if you have a circular mRNA, when you stop translating, so the ribosome goes to the stop codon, maybe it's easier for the ribosomes to recirculate on the mRNA, but nobody really knows if that's true or not. Okay, so in virus-infected cells, this basic system has been modified and expanded for a variety of reasons, and we're going to talk about that now. And one of these types of expansions is called ribosome shunting. Now, basically what viruses will try and do is to take over the entire translational apparatus of the cell. So as you will see, they will try and shut off the translation of host mRNAs. They want to have the ribosomes and the tRNAs all dedicated to making their proteins, viral proteins. So I will talk about some ways that that happens. But in turn, the viruses have to have a way to escape their own inhibition of translation. And this is one of them. So that's why we're talking about modifications of the translation apparatus to make the viruses have the system dedicated to them, but also not to be inhibited by their own actions. So ribosome shunting is an interesting mechanism whereby we think that ribosomes bind the cap, just as I've told you. This happens on viral mRNAs. They bind the cap and then they pass over most of the 5' UTR. Okay, so that is shown by this experiment where we have made a messenger RNA. It's capped, and it has a lot of secondary structure in the UTR. And this secondary structure, of course, would have to be melted out in order to get translation of the protein. And this is a Western blot on the right where we're looking for the protein that is made by translation of this mRNA. So if you take this wild-type mRNA and you put it in a cell or in a translation extract, you get proteins made. Now, if you put a very stable stem loop structure right here, right next to the AUG, that's shown in red. So this is a stem loop, which is so stable it will for sure block the movement of ribosomes and prevent them from getting to the AUG. We know this. This has no effect in this position on protein synthesis. So somehow the ribosomes are getting around it. If you put the same stem loop here at near the 5' end, it blocks translation of this mRNA. All right, so this shows you that the stem loop in itself is enough to block the passage of the ribosome here only. So we think that the ribosomes begin to scan, and they can scan past this region, and this blocks it. So they normally would scan through here. And then at some point in here, they are released or transported in some way directly to the AUG. I will show you a model for that in a moment. So that's why it's called shunting. The ribosomes are shunted from one part of this UTR to the other. So they can shunt over this stem loop, but they can't apparently shunt over this one. So that's the evidence for our ribosome shunting. And this is a model for what we think happens. The ribosomes bind at the 5' end. They scan a bit, and then they reach some secondary structure which can then hand them off directly to the AUG. We really don't know how this works. This is fanciful drawing. It's kind of a direct translocation which doesn't require scanning. So putting in a stem loop structure doesn't affect this. Nevertheless, these stem loop structures are important for the shunting. Apparently the ribosomes bind to them and are transferred in some way. So this is observed to occur in the mRNAs of a couple of different viruses, a plant virus, an adenovirus, and Sendai virus. And we think one of the reasons why it occurs, it's predicted to decrease the dependence of mRNAs for EIF4F. Now, EIF4F is the whole complex of EIF4A, 4G, and 4E at the 5' end of the mRNA. And remember, if you have a lot of secondary structure in mRNA, you need to have that EIF4A helicase. Now, the shunting is predicted to get around the need for melting. So the virus could in some way inhibit this EIF4F complex so that cellular messages are not translated. Yet its messages can be translated because they have this shunting mechanism that allows them to get over secondary structure. So that's ribosome shunting. Another different way of translating is internal initiation. And this is, as I said, first discovered for poliovirus RNAs. When the poliovirus genome was sequenced, it was found there was a very long 5' non-coding region at the 5' end. So this is 740 nucleotides, longer than most 5' UTRs that had been discovered up to that time. It was about 1980 or so. And it wasn't clear how the ribosomes would get through this because this is full of AUG codons, which would impair ribosome scanning. So in addition, what made this RNA complex, remember this is an mRNA, this is a plus-strand RNA genome, it's also an mRNA. There was not a cap at the 5' end. It had a little protein linked to it, which, if you remember from our talk on RNA synthesis, is the primer for RNA initiation. So there's no cap. How do the ribosomes attach to this RNA, and how do they get through all of these AUGs? Well, someone had the idea that maybe this allows ribosomes to bind internally. So they did this experiment. They made two different mRNAs, and they both have two protein coding regions. So one here in orange and one here in blue. So you're making orange proteins here and blue proteins here. Now, this is a capped mRNA. If you put this in a cell, you make a lot of orange protein. But remember, eukaryotic mRNAs are not very good at making more than one protein. So most of the ribosomes will fall off after making the orange protein, and very few will make it to the second protein coding region. It's just a fact of the way eukaryotic mRNAs work. So we have one blue protein here to illustrate that not many of those are made. if you take this sequence from the 5' end of poliovirus RNA, that sequence here, this 740-some base sequence, you make a DNA copy of it, you put it into this construct, you make it RNA, of course, eventually. This is a RNA. So that's this sequence here in the middle, which we've labeled IRES. And then you put this into a cell. Now you can make a lot of orange proteins as well as a lot of blue proteins. So somehow this sequence enables the ribosomes to get inside the mRNA and initiate translation. And it was called an internal ribosome entry site, or IRES. That's where the name comes from. Now on the bottom is another experiment that was done. This was done, instead of putting these RNAs into regular uninfected cells, they were put into poliovirus-infected cells. Now, as we will see later, in poliovirus-infected cells, the 5' end-dependent translation is completely inhibited. So ribosomes cannot get onto the 5' end. They're blocked at loading onto the cap. We'll talk about the mechanism. So this mRNA is absolutely not translated at all. No orange proteins are made, no blue proteins, because the ribosomes can't load. This is a poliovirus-infected cell. But if you take this RNA where you have an iris, you make plenty of blue proteins, because the ribosomes don't need a 5' end, which is blocked by polio infection, to translate the blue protein. They can load internally, which is not affected by the block at the 5' end. So this showed that internal ribosome entry actually bypasses a 5' end. That's why I like to call the other mechanism 5' end dependent, because this one is not. You don't need a free 5' end. So internal ribosome entry site was coined to describe this. Now, this predicts that a 5' end is not needed, but it doesn't prove it. But the following very cool experiment does prove that. So if you want to prove that a 5' end is needed for 5' end dependent translation, but not iris dependent translation, you make a circular mRNA. And you can do this by ligating the ends together. You have to make a little oligonucleotide that bridges the ends, and then you can ligate them together, and you make circles like this. And in this experiment, we have two circular mRNAs. One has an iris, and the other doesn't. And this has an AUG, so this is the open reading frame for a protein, and this one has the same open reading frame, but no iris. This RNA will make protein when you try to translate it, because the ribosomes can bind to the iris. This one has no iris. The ribosomes can't recognize it. There's no free 5' end with a cap, so they can't translate it. So this shows that an iris doesn't need a 5' end in order for translation to occur. And what does an iris look like? Or what color is an iris? Irises are blue usually. These are what irises look like. These are from a variety of different viruses. This is poliovirus iris here, the one that was originally studied. This is another virus related to poliovirus, encephalomyocarditis virus. Here is the iris of hepatitis C virus, a virus in a different family entirely. And then here is the iris of an insect picornavirus. So polio and EMCV are all picornaviruses, as is this one. So you see, first of all, the yellow parts are the iris part, here, 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 and here. They're all different. The sequences are completely different. And even the structures are different. So you actually cannot look at a nucleotide sequence and say, aha, that's an iris, because there's no signature. The structure is very important for the ribosomes to be able to bind, but it's very hard to scan for such structures when you're looking at sequences. So for all of these irises, the ribosome will bind internally, very close to the AUG, and initiate translation, and a 5' end is not needed. You could circularize all of these RNAs and they would still be translated. Let's talk a little bit about how these work, and we'll start with the iris of hepatitis C virus, which is one of the simplest. So hepatitis C virus is in the flavivirus family, and other members of that family would be West Nile virus. And this virus has an RNA genome, which is shown here. It has an iris at the 5' end, and the ribosome, the 40S subunit, can bind directly to this highly structured iris. Remember here, this is the hepatitis C virus iris, panel C. It's highly structured. The ribosome will bind it. If you take purified hepatitis C virus RNA and purified 40S subunits and mix them, they will bind. So this RNA has evolved to be able to bind directly the 40S subunit. So the 40S binds the iris without any proteins, and that places the 40S subunit right on top of the initiation codon. And then EIF3 can come in, and the big subunit then comes in as well. So this is an example of a very simple RNA-protein interaction. The RNA of the virus can interact directly with the proteins and RNAs of the ribosome. Now, for other irises, it's probably a little more complex. Here is the Hep C iris for reference. Just the ribosomal 40S subunit can bind the iris. This is an iris of, say, picornaviruses. Here we think more of the initiation proteins are needed for binding. In this case, we believe that EIF4G binds to the iris, which then, if you remember, binds the EIF3 and then the 40S subunit. So it's a little different from the Hep C example where the 40S can bind the iris directly. Here, it has to engage EIF4G. Now, why the N-terminus is broken here, you will see a bit later. That is an interesting question. So this is interesting compared to cap-dependent translation or 5' end-dependent translation. Remember, the cap is essential for bringing the complex to the 5' end. You don't need a cap for with an iris mRNA. The ribosome can be brought by direct protein-RNA interactions. Now, since these viral irises were discovered, many, many more were discovered in viruses. And on the left is a list of viral irises. So we have them in many different virus families. I told you about picorna and flavivirus irises. There are also viruses in the pestiviruses. Retroviruses have irises as well, as do the insect picorna-like viruses. And we also have found irises in cellular mRNAs and a variety of genes which are shown here. And this we don't quite understand because these mRNAs that have these irises also have a cap at their 5' end. And it may be that at certain times in the cell cycle, 5' end-dependent translation is inhibited. We know this occurs during mitosis, for example, and it could be that if you need certain proteins made during that time, you put an iris in them, and they can continue to be made. But proving that has been a little difficult. All right, let's go back to the initiation step now. You remember we went through the whole initiation step of 5' end-dependent initiation, ending up with the MET tRNA in the P site, ready for translation. There are very interesting examples among viruses of methionine-independent translation. So here we have a MET tRNA sitting down on the AUG. So the ribosome has scanned down the mRNA, looks for the first AUG. As soon as it finds it, it puts a tRNA there. There are examples of methionine-independent in not only cells, but in virus-infected cells. And here are two of them. This is an example where you don't need a methionine tRNA because the viral RNA folds in a way that mimics a tRNA. So this is a viral RNA. It happens to be a plus-strand RNA. We'll talk about it in a moment. The very 5' end is folding in a way that looks like a MET tRNA. So not a MET tRNA, but an initiator tRNA. This happens to be initiating at a proline triplet, although there is no amino acid placed in here because this is not charged with an amino acid. All right, so this RNA can initiate translation without a MET tRNA. The first amino acid to go in this case, is an alanine in the A site here. Right? Again, because the RNA is folding to look like a tRNA, even though there's no amino acid attached to it. So that's one example, and there's another one. This is a plant viral RNA where the 3' end has a tRNA-like structure. This is actually charged with valine. You can see the valine amino acid is attached here. So this sits down on a valine codon, and the first amino acid to be put in is a valine. So the RNA 3' end mimics a methionine initiator, instead putting in valine. We're not sure why they do this at all. Now, the RNA genome of cricket paralysis virus is shown here. And this is, as far as I know, the only eukaryotic viral mRNA where you have actually two open reading frames separated by an iris. Now, an iris would, in theory, give you the ability to make multiple proteins from one messenger RNA, right? So you could have 5' end, you could have one open reading frame, you could have an iris, a second open reading frame, an iris, a third open reading frame. You could be like a bacteria, which can do polycistronic mRNAs. But we don't see that at all. I don't know of any mRNAs in eukaryotic cells where that happens except for this one. So this is cricket paralysis virus. It is a picornavirus. It paralyzes crickets. That's how it was discovered in Australia. The farmers came out one morning and saw paralyzed crickets on the ground, and they sent them to a virologist, and the virologist identified this virus. So people are trying to figure out if it can be used for pest control. The interesting thing about the virus, when sequenced, it would look almost like polio, except instead of one long open reading frame, it was split in two, and there' an iris in between here labeled IGR, intergenic region. So this is an iris splitting two open reading frames. There's also an iris at the 5' end and VPG, just like the picornaviruses. Now, this iris here looks like this, and in fact, this is a sequence that can mimic a tRNA. So that's the one I just showed you before. The way initiation works on this iris is that the ribosome binds internally, and the RNA mimics the tRNA so that initiation begins without a methionine codon. But the interesting point here, well, there are two interesting points. This is very interesting, the methionine-independent initiation, but also the fact that you have, this is a polycistronic mRNA, which only happens in bacteria, and is the only example that I know of that. So here's the problem, and we're going to talk about other ways to get around this. So eukaryotic mRNAs are typically monocistronic, one mRNA, one protein. Bacterial and archaeal mRNAs are polycistronic. You can make several proteins from one messenger RNA. And that's because at the 5' end, around the AUG of each of these ORFs, there's a Shine-Delgarno sequence, which is complementary to a sequence on the ribosome, ribosomal RNA. So the ribosomes can bind internally on these bacterial mRNAs. Most mRNAs in eukaryotic cells can't do that, with the exception of that cricket paralysis virus. I don't know why they don't do it, because you could put irises into these mRNAs and make multiple proteins. In fact, we do that in the lab all the time. If we want to make two proteins from one mRNA in a mammalian cell, we just put an iris. You can buy irises from companies just for that purpose. Make two proteins from one mRNA, but they are not naturally occurring. All right, so this problem, this monocistronic limitation, is gotten around by many viruses, because, and we've mentioned this before, many viruses, there's one RNA genome. You have to be able to make multiple proteins. So I want to tell you now the different ways that viruses get around that, or I call it maximizing the coding capacity of the viral genome. So here are some of the strategies we're going to talk about, and some examples of the virus families that utilize them. We've talked before about the polyprotein strategy. I just told you about internal initiation. We'll also talk about leaky scanning, reinitiation, suppression of termination, and frame shifting. So these are all very interesting ways to get more than one protein from a message in a eukaryotic cell, and they're all discovered with viruses. And some of them happen in uninfected cells as well. Polyprotein synthesis you have seen. Coronaviruses make a polyprotein and chop it up with proteinases. That way you can get a dozen proteins from one mRNA, so that's obvious. There are other plus-strand viruses. The toga viruses do the same thing. They make a polyprotein and process it. So different viral families do that. Internal ribosome entry. We talked about the RNA genome of cricket paralysis virus making two proteins by putting an iris in between them. And that's the only example I know of this. These two proteins, by the way, are in fact polyproteins on their own. So these are also processed to give more proteins. Leaky scanning. This occurs in the genome of paramyxoviruses, which are negative-stranded RNA viruses with an envelope. And here is an interesting example. These These viruses make subgenomic mRNAs. They make 7 or 8 subgenomic mRNAs from the negative strand RNA genome. And we're looking here at one of those mRNAs that encodes for, this one mRNA encodes for 1, 2, 3, 4, 5, 6, 7 different proteins by this interesting leaky scanning mechanism. Actually, two of the proteins are made by RNA editing. So you remember RNA editing is when you make the mRNA, you insert an extra base during transcription, which is not templated. So you shift the reading frame so you can make this protein and this protein, which have different C termini. But the C proteins, 1, 2, 3, 4, are all made by leaky scanning. So let's see how this works. So we have a cap at the 5' end. The ribosome binds, and it starts to scan down, and it reaches this ACG codon. Okay? Now, ACG can be an initiator. tRNAs will bind to that, but it's not very efficient. So maybe ribosomes start translating only 5% of the time. So they make this C' protein, but a number of ribosomes will keep scanning without initiating. And then they reach this AUG at 104. Now, that's an AUG codon, so ribosomes like that. But you know, the surrounding sequence around an AUG is very important for the efficiency, and we call that the context of the AUG. And this AUG has a really bad context. So again, a few ribosomes translate and make this P protein, but many of them continue to scan. Then they reach this AUG at 114, which is an AUG, and it has a great context. Yes?
Since I guess they make multiple proteins, I mean, are these proteins functional? Because they just kind of like, I guess, produce them in different places?
Sure. Good question. I mean, this would be uninteresting if they weren't functional, right? Yes, you can do experiments to eliminate each of these proteins, and they're important. So they differ at their end termini. And in fact, this one's in a totally different reading frame, the P protein. That's why it's brown here. So this is a totally different protein from the others, which just differ by the length of their end terminus. But they do have important functions.
I think all certain amino acids would really affect the functionality of the
Yeah.
cell.
Yeah. Yeah.
Why would it pass over AUG 104?
So the context is bad. Around the AUG, there are a couple of bases on either side which have to be a certain sequence to get maximal initiation. It's called the consensus sequence. And if in eukaryotic mRNAs, there are really no perfect consensus mRNAs because every mRNA doesn't have to be translated to its maximal efficiency. So that AUG at 104 has a bad consensus surrounding the AUG. And then we have, so these are leaky scanning, 81, 104, and 114. These two proteins are RNA editing. And then these two here are very interesting, AUG 183 and 201. These are made by ribosome shunting. Remember that mechanism where the ribosomes jump a long distance? That's what they do here. They start to scan, and then they get past AUG 114, and a number of them are still scanning, and they jump to 181 or 175. So it's amazing. From one mRNA, a virus has evolved to be able to make multiple proteins. Yeah?
I guess when you're talking about the first three, is the kind of bad context or like bad starting sequence
Right.
related to how necessary the protein is for the virus? Or is it 114?
So the context in this and the codon, the nature of the codon, just allows more ribosomes to pass over it so that you can get the downstream protein made.
And then is that one really more important since you're wanting to skip the others, or it's just the way it
Well, apparently you need to make all of them,
Yeah.
right? But you don't need all of them at the same levels. And this just ensures you that eventually you get to the downstream one, right? So those are the three made by leaky scanning. And then the others are different mechanisms as well. All right. What else do we have? Reinitiation of translation. So on most cellular messenger RNAs, I told you, you have an open reading frame, you make one protein. But there are a couple of examples of viral RNAs where there are some short open reading frames actually within what's called the non-translated region. Obviously, this is not technically a non-translated region, but it would normally look like one because it's upstream of the main open reading frame. But in some virus-infected cells, you can make these short proteins. They're called upstream open reading frames. And the ribosomes keep going, and then they reinitiate at this downstream ORF. So you can make 2 proteins from one mRNA. That's one example of reinitiation. The second one is an influenza virus mRNA, which can code for 2 proteins called M1 and BM2. What happens here is the ribosomes initiate here. They make this protein. UAA is a termination codon, so they stop. But look, the UAA is part of the next AUG. So they reinitiate immediately. So normally, after terminations, ribosomes move a little more and then fall off, but there's nowhere to go here. They move, they see an AUG, and boom, they start translating again. So that's another kind of reinitiation. Yeah?
But this shows that the open reading frames overlap, so does the ribosome kind of move backwards before?
No, it doesn't in this case. It's just, that's the actual open reading frame, but the protein begins right here. Yeah. These don't. But there is an example of that, which we will get to, overlapping reading frames. Reinitiation. Then we have suppression of termination. So protein synthesis stops when the ribosome reads a termination codon. Now, how does that work? So you make proteins by adding charged tRNAs to the A site, and then a peptide bond is formed, and here's the growing peptide chain. And then you throw away the tRNA without the amino acid. So this one has just been linked to the growing peptide chain. If this were a triplet for the next amino acid, a new tRNA would come in here, and then the amino acid would be linked up to this chain. But this is a termination codon, UAA. It is recognized by termination factors, ERF1 and ERF3, and those are shown here. These factors basically look like a tRNA, but they sit in the termination site. And there's no tRNA there, there's no amino acid, so that halts translation. So that's how termination normally happens. You have these termination factors recognizing the termination codon, remember there are 3 of them, and termination and translation stops. But sometimes these stop codons can be recognized by a tRNA that has an amino acid on it. And that's called suppression of termination, and obviously allows translation to continue. And that happens in a number of viral genomes. Here is one example. It is a retroviral genome, and here is the mRNA made by this retrovirus. Retroviruses encode a GAG protein at their 5' end. This is the structural protein that forms the core, and a Pol protein, which has reverse transcriptase and RNase H and integrase. In this particular retrovirus, there is a stop codon between the GAG and the Pol coding regions, right here, UAG. So ribosomes begin to translate this mRNA, they make GAG, and they stop. How do you make polymerase? If you don't make polymerase, you're never going to reverse transcribe. Well, right downstream of this UAG, there's this very interesting structure. It's called a pseudoknot. It's a stem loop where the unpaired bases in the loop base pair with a downstream sequence. I showed a picture of this some time ago. And apparently this causes the ribosomes to pause. The ribosomes hit this structure and they pause, and that gives enough time for a tRNA to actually sit into this UAG site and put an amino acid there. So this can be a misreading of the codon, which is usually what happens. It's not quite the right codon going in there, but it's close enough and you get an amino acid put in. So that's one example. There's also suppression in the alpha viruses. These viruses, if you remember, have a plus-strand RNA genome. When they are translated, initially they make a precursor, a polyprotein precursor called P123. It stops right here. There is a termination codon. But what's beyond it is NSP4, which is the viral RNA polymerase. So it has to make this. So there is some suppression of this termination codon. Now, because both the polymerase and this RNA polymerase are enzymes, you don't need to have a lot of them because they're catalytic. So this suppression only happens about 5 to 10% of the time that the ribosome comes down, because you don't need 100% translation because they're not structural proteins. So this works. Gives you a little bit of enzyme made, and that's how the virus accesses this coding sequence. So there are two kinds of suppressions. There are specific suppressor tRNAs that recognize these 3 termination codons, and they put in a specific amino acid. So, for example, there is a suppressor tRNA that will recognize the terminator UGA. It puts selenocysteine in, a rare amino acid. And then there are normal tRNAs that misread termination codons, which is what I told you about. Then we have ribosomal frame shifting. So to get back to your question, when you have overlapping reading frames, on another retrovirus, the mRNA, again, encodes GAG and POL. Remember, in the retrovirus I just showed you, the GAG and POL were separated by a terminator. Here, the GAG and POL are in 2 different reading frames, which overlap. So how do you get both proteins made? Well, you get, in fact, in infected cells, you get GAG made, and you get a GAG-POL fusion. And the GAG-POL fusion can be cleaved by a protease to release the polymerase. This is made by ribosomal frame shifting. Basically, the ribosomes, 5 to 10% of the time, they reach the end of GAG, they back up a base, and shift to another reading frame. So they shift, in this case, to the -1 reading frame, and now they can translate the POL gene, which is in the -1 frame with respect to the GAG. That's why it's called ribosomal frame shifting. Okay? That's another way to make a protein that you normally can't access. And here's how this happens. These frame shiftings usually happen at what's called a slippery sequence. It has a lot of A's and U's, so the ribosome gets somewhat confused. So here you have our normal reading of this triplet, this UUA, but now the tRNA slips back 1. So we've gone here to -1. We're back 1. So now you see this this U is base paired with this A here. And then the next triplet to be recognized is going to be AUA instead of UAG, which in this case would be a terminator. So now you start and you continue the protein. So the next codon that will be read is not a terminator. It's an insertion of an amino acid. So you've slipped back to the -1 frame, and now you can access the rest of that open reading frame. So it's a very simple -1 slippage right here. Ribosomal frame shifting. Okay, so those are a couple of ways that viruses can make many proteins from one messenger RNA. Now, the other issue I wanted to talk about is the regulation of translation in virus-infected cells. And as I said, viruses want the entire machinery dedicated to them. They don't want any cell proteins made. And here's one example of how viruses achieve that. So this is the initiation step which we talked about. Here is the 40S subunit attached to the RNA, and here's the ternary complex, the MET tRNA, EIF2 GTP, and now that MET tRNA is sitting down on the AUG codon. So remember I told you at that step, GTP is hydrolyzed to GDP and EIF2 is released. All right? This is a very important factor, this EIF2, because what it has to do eventually is pick up more MET tRNA and do another round of initiation. But in order to do this, to pick up another tRNA and make a ternary complex, the GDP has to be exchanged for a GTP, because GDP won't work, can't be hydrolyzed to release the energy that's needed. So there's a second protein called EIF2B, which binds this EIF2 GDP and exchanges the GDP for GTP. So it's basically a guanine exchange factor. Now we have EIF2 with GTP, and it can go through this whole sequence again. Okay, now in cells there is a mechanism to regulate this pathway, and it involves phosphorylation of EIF2. We'll talk about how that happens in a moment. Basically, EIF2 is phosphorylated, you see right here, under certain conditions. When that happens, it binds very tightly to EIF2B, and it doesn't get released at this step. So you eventually soak up all the EIF2B in the cell, which happens to be limiting, and you can't get any more recycling of this EIF2, and translation stops. All right? So in fact, this is a response of many cells to virus infection. They phosphorylate EIF2 with the idea of halting virus infection and maybe preventing spread of the virus to other cells. And that phosphorylation of EIF2 is carried out by a number of different protein kinases in cells. They're called EIF2 alpha kinases. One of them is called PKR, another is called PERK, and another is called GCN2. And they all have this kinase domain which is involved in phosphorylating EIF2. Now we're going to talk about PKR and PERK today. Now the PKR response is an antiviral response of the cell. So on the left here we have a cell being infected by a virus. The cell senses that it's been infected. How it does so we'll talk about later on. And one of the responses to the presence of virus in the cell is that the cell makes interferon. Interferon is a protein, a series of proteins that are involved in establishing an antiviral state in cells. These interferon molecules that are made come out of the cell and they bind to interferon receptors on the cell surface. And they then induce the synthesis of a number of proteins that have antiviral properties. And one of those proteins is PKR, which is the kinase that phosphorylates EIF2. And so when cells are infected, the cells respond by producing this protein, which is engineered to stop translation and stop the infection. Now, the way that this works is that PKR is normally present at a very low level. When it is produced, it needs to be activated. So the mRNA is induced by interferon. It has to be activated before it can phosphorylate EIF2. And what we think happens is when viruses infect cells, most of them produce double-stranded RNA. The double-stranded RNA will bind to PKR, which is shown here, this purple, orange, brown molecule. This is PKR. Multiple molecules of PKR bind the RNA, and they phosphorylate each other, and that activates the kinase. So PKR is not active as it's produced. It has to bind double-stranded RNA in order to be activated so that it can then go on to phosphorylate EIF2. So PKR is induced and activated by virus infected. It is induced by interferon, and it is activated by the presence of double-stranded RNA. And its activation leads to the inhibition of host translation. It is also an activator of apoptosis. Now, viruses don't like this, right? They don't want translation to be shut down because they cannot make their viral proteins. So they have evolved, almost every virus has evolved some mechanism to evade this PKR pathway. So that tells you right away how important this is. I'm going to show you one of them, and that's a small RNA made by adenovirus. This is a big double-stranded DNA virus. This RNA is called VA1 RNA. It's rather short, and its function is to prevent activation of PKR. So what happens is, again, remember, PKR is induced by virus infection. It binds double-stranded RNA, which is produced in virus-infected cells. In an adenovirus-infected cell, it would be produced when you get transcription of both strands of DNA, and the transcripts would be complementary. They would hybridize and make double-stranded RNA. This binds the kinase and activates it. You inhibit protein synthesis. What adenovirus does, it makes this small RNA called VA-RNA1, which binds one molecule of PKR, not more than one. So PKR cannot activate itself. It cannot phosphorylate itself. It cannot phosphorylate EIF2, so you don't get inhibition of protein synthesis. So that is a function of this antagonist to prevent the PKR inhibition of protein synthesis. Other viruses take different approaches to this. Here is an example of vaccinia virus. It makes a pseudo-substrate that mimics EIF2 alpha. So here's the structure of EIF2 alpha. Remember, this is the substrate for PKR. This is the protein that PKR phosphorylates. Then that results in translation inhibition. It's normally phosphorylated here on serine 51. Vaccinia virus makes two proteins in very large quantities, which are mimics of EIF2 alpha. And these basically take up all the PKR that is produced, so the PKR never gets to phosphorylate EIF2 alpha. Rather, the PKR works on these K3L and M156R proteins and never gets to phosphorylate EIF2 alpha. So those are just two examples, and these are examples of how different viruses antagonize this pathway, this EIF2 alpha phosphorylation pathway. You have herpes, influenza, adenovirus, baculos, hep C. Amazing. And they're all different mechanisms. They have different targets. But the end result is that they don't want EIF2 alpha phosphorylated. So obviously this is a very important antiviral effect, and viruses have evolved a number of ways to get around it. Now there's another EIF2 kinase in cells called PERK. That stands for PKR-like ER transmembrane kinase. ER is endoplasmic reticulum. PKR was the first EIF2 alpha kinase discovered. This was subsequently found, and it has homology to PKR, hence the name. So what happens here is that this kinase responds to misfolded proteins in the endoplasmic reticulum. Now PKR, remember, responds to double-stranded RNA. PERK responds to misfolded proteins, which often happens in virus-infected cells. It happens at other times as well. If you take away oxygen from cells or glucose, mutations, toxins, virus-infected, these all cause misfolding of proteins in the ER. This triggers what's called the unfolded protein response. Now, the unfolded protein response is a series of proteins and signal transduction pathways which says, there are misfolded proteins here. We have to stop making proteins until we can sort out what is going on. All right? We don't want to make more proteins until we can stop the misfolding. So that the UPR shuts down translation. How does it do it? By phosphorylation of EIF2 alpha via this kinase PERK. And so in many virus-infected cells, this happens as well. So here's the pathway. You have our ER. Normally, you're making nicely folded proteins in the ER. You have some kind of stress, a toxin, a virus infection. The proteins become misfolded. They're not in their proper structure. There's a protein in the lumen of the ER called BIP. Stands for binding protein. It will bind to misfolded proteins. And as BIP binds to more and more misfolded proteins, it's taken away from PERK. PERK is a transmembrane protein in the ER. It's normally inactive when bound to BIP. When BIP is titrated away because BIP is binding the misfolded proteins, PERK becomes active, it dimerizes, it phosphorylates itself, and then it can be active and go on to phosphorylate EIF2 alpha. And this is another way that viruses are responding to, cells are responding to virus infection to shut down translation. And again, viruses have evolved ways to get around this as well. But cells do other things as well. We're just beginning to learn these. When they are stressed, not only do they activate this unfolded protein response, but they shut off translation by putting mRNAs into what are called stress granules. So here's a nicely translating mRNA. You see the flying turkeys. These are ribosomes are coming around the mRNA. Don't these look like turkeys, right? They're flying turkeys. They're coming around translating. It's a circularized mRNA. You put some stress on the cell. You infect it. You put a toxin on. The cell has a mechanism to shunt these into these stress granules, which are very specific collections in the cytoplasm. They have these mRNAs, and they have a variety of proteins that make up the granules. So you can actually see these stress granules in the cytoplasm by staining for these proteins with antibodies. And these are stalled mRNAs. The ribosomes are frozen on the mRNA. They're not making protein. So the cell is saying, again, we're stressed. We don't want to make proteins. It's a waste of energy. So let's stop translation until we can sort this out. And they put them in stress granules. So a virus infects a cell. Its mRNAs get shunted into stress granules. This is not a good thing, right? It's not good for the mRNA to be shunted in there. So what do viruses do? They antagonize the formation of stress granules. Here, West Nile virus and dengue virus antagonize the incorporation of two structural proteins into the stress granule, thereby blocking their production. And poliovirus, one of its proteinases, 3C, cleaves one of these components, G3BP, of the stress granule. So it prevents the formation of these granules so that its mRNAs are not incorporated into them, and it can continue to be translated. Another level of regulation of translation is at EIF4F, and now we get to talk about why the red part at the end of EIF4G is broken. So remember, the ribosome is brought to the mRNA by this complex, which is called EIF4F. This protein, EIF4G, is cleaved in cells infected with a variety of picornaviruses. It's cleaved by viral proteinases right here, and that prevents this from translating a capped mRNA. But because these viruses have an iris, they don't need a cap, so they don't care. And in fact, 4G still can bind the iris, but it just doesn't bind EIF4E. And if the RNA doesn't have a cap, that doesn't matter. So in poliovirus-infected cells, at different times after infection, if we look at the rate of protein synthesis, you see it declines initially very rapidly. So this is inhibition of cell translation, and then it is replaced with viral protein synthesis. And then the cells die. The shutoff is also shown here. This is a polyacrylamide gel where the proteins are labeled with S35 methionine. You can see at time 0 after infection, there are a lot of cell proteins being made, also at time 1. But look, at hour 3, you start to see an inhibition of translation. Now at hour 5, most of the cell proteins are no longer made. They are replaced by viral proteins, shown here. So that is what is happening in virus-infected cells. The cell translation is shut down so that all of the machinery can be devoted to translating viral mRNAs. And this is an experiment that shows you the basis for that inhibition. So on the left is the same experiment I just showed you. It's a time course of polio infection, and we're labeling proteins with S35 methionine. So you can see in this experiment, shutoff starts to occur around 2 hours and 15 minutes post-infection. And it's all host is all shut off pretty much by 2 3/4 hours. Okay? And these two are Western blots using an antibody against EIF4G. Remember, EIF4G is that linker protein that is cleaved in picorna-infected cells. There are two kinds of EIF4G in cells, EIF4G1 and 2. And you can see they are both cleaved during infection. This one is cleaved starting at about 1 hour and 15 minutes. This one is cleaved around 2 hours and 15 minutes. So actually the cleavage of 4G2 really correlates very nicely with the shutoff. So again, that's the protein that it is cleaved right here near the N-terminus, so cell messages can't be translated because the ribosome can't be recruited to the capped mRNAs. So that's the basis for that. So again, it's shown diagrammatically here. EIF4G is cleaved right here by a viral protease. That separates the EIF4G from the rest of the molecule, so it can't support 5' end-dependent translation. But this fragment here, the C-terminal fragment of 4G, is perfectly good to bind irises. So that's why the virus translation continues to work even when this 4G is cleaved. And the last thing I want to tell you about is something relatively new for viruses. And this is a very exciting area of biology in general, microRNAs, which is part of this whole RNA interference pathway in the cells. There are many different kinds of small RNAs, which we now know are not only important for antiviral responses, but for regulating gene expression. So microRNAs are a subset of that whole RNA interference pathway. These are very small RNAs. They're typically non-coding. They can be encoded by viruses or by the cell. And they're transcribed by Pol2 or 3 into 60 to 70 nucleotide transcripts that are then processed by enzymes to 21 nucleotide structures, which then have activity. All right? Now, in the cell, these are incredibly important for regulating cell genes. They control gene expression post-transcriptionally by either repressing translation or degrading the mRNA. And whether these microRNAs do one or the other depends on the complementarity of sequences within the 3' non-coding region of the messenger RNA. I'll show you that in a moment. So these microRNAs function to inhibit mRNAs in a protein complex. They're called microRNPs. They have a couple of cellular proteins here, as you can see. And again, the complementarity to the 3' UTR of the message depends whether the message is degraded or translation is repressed. And here's a schematic of an interaction of a typical microRNA with the 3' UTR. So here's the end of the open reading frame. Our termination codon would be here. Usually they're binding about more than 15 nucleotides from the end of the coding region. There is typically homology on either side of a bulge. This is how the microRNAs work. And then the extent of complementarity here controls whether this interaction leads to degradation of the mRNA or inhibition of translation. This is actually a very active area of work right now to sort out how this works. So these are the two ways that microRNAs can regulate translation, which is what we've been talking about today. Remember, 5' end-dependent translation depends on a ribosome being brought to the mRNA via this Cap4E interaction. So there are some data which suggests that the formation of a microRNP at the 3' non-coding end inhibits this joining at the 5' end, either at the cap binding step or the 60S joining stage. Now you can see that this is possible because if in fact the RNAs are circularized as we suggested, that would bring the microRNAs down to the 3' end, to the 5' end. So that would repress initiation. Here, there's also some idea that the microRNPs can actually affect elongation. We haven't talked about the regulation of the elongation step at all, just the initiation step, but there is regulation at that step, and perhaps microRNAs cause the ribosomes to drop off at a higher rate. So that's how they might impact translation. And then, of course, the other part, which we're not talking about, is that they would degrade the mRNAs directly. So I just want to leave you with a couple of examples of how microRNAs play into virus infections. And again, remember, there are both viral mRNAs and there are cellular microRNAs, which have different roles. So here's an example of a cellular microRNA. It's called miR-122. It's made only in the liver in us, and it's required for hepatitis C virus replication. So hep C is hepatotropic, doesn't replicate anywhere else as far as we know. And this is probably because miR is a liver-specific microRNA. And this miR binds the 5' non-coding region of hep C very close to the iris and somehow allows the virus to replicate. People are just trying to sort out how that works. So no miR, no hep C replication. So guess what? Companies are trying to design ways to target miR-122 as an anti-hep C therapy. You can do that. You can design complementary RNAs that will bind the miR and take it out of circulation. There are also microRNAs in the cell that are antiviral, and they can either target viral genes, so you can have a cellular miR that would inhibit the production of a viral protein and inhibit viral replication, or in many cases there are specific cellular genes that are needed for virus replication. So the miRs, which are cellular miRs, are actually reducing the expression of those cellular genes that are needed for the virus, and that reduces virus replication. So those are cellular miRs that have some role. We have a positive effect on virus replication, and some miRs inhibit virus replication. And then we have virus-encoded microRNAs. And so far we have slightly more than 200 that have been identified in these 6 different virus families. A lot of them in the herpes viruses, as you can see. Some controversy about whether they really exist in retroviruses or not. But these appear to have a variety of effects during infection. Many of them block apoptosis. So programmed cell death is a response of cells to virus infection. The cell wants to die so that viruses cannot be produced. So there are many, and the virus, of course, doesn't like that. Especially viruses with long replication cycles, the virus does not want apoptosis to occur. So these miRs block apoptosis. That is their function. They're produced from the virus genome, and they block apoptosis. There are also examples of miRs that block immune responses, which we may hear about from Dr. Silverstein. Many cells go into cell cycle arrest when they're infected. And remember, DNA viruses need that DNA replication machinery to be active, so the miRs prevent the cell cycle arrest. And many others promote latency of viruses, the ability to remain with the cell for long periods. So these are pretty new areas of research, and this is going to be explored really in an interesting way in coming years, how these viral and cellular mirrors have an interplay and how they result in either an infection or not. And this is something we didn't even know about 5 years ago.